Hello, my name is Michelle Anya Sankoli, and the theme I've chosen for my bio video project is the gut microbiomes. I want to begin my project with a short abstract on what gut microbiomes actually are, their mechanism and their role, and in my own few words, I want to describe them as a diverse community of microorganisms that live in our digestive system. They help with digestion, with nutrient absorption, they support our immune system, and they also, as microbes, interact with our gut cells, affecting processes such as cell cycling, which can impact our overall health. While this is a very wide title with a range of different themes to talk about, I have chosen four main topics to focus on that revolve around cell cycling and how gut microbiomes impact it. So the first topic I've chosen is the role of gut microbiota in host cell cycling, where we'll be exploring specific bacterial species or strains known to influence cell cycling processes, as well as discuss how microbial-derived metabolites regulate cell cycling machinery in intestinal epithelial cells. It's very important to highlight the importance of maintaining a symbiotic relationship between the gut microbiota and the host for optimal cell cycling regulation as well as gut health. The second topic I've chosen to discuss is the emerging techniques for studying gut microbiome and host interactions. Here we will describe innovative experimental models such as gnotobiotic mice for investigating the role of gut microbiota in cell cycling and as well as discuss advancements in multi-omics approaches. Then we move on to speak about the impact of dysbiosis on gut cell cycling and disease. And here we will actually be talking about how dysbiosis can perturb host cell cycling and contribute to disease pathogenesis. I'll be providing some examples of some diseases or conditions associated with dysbiosis, but I'll be mainly focusing on inflammatory bowel disease. And finally, the last topic I will be talking about is a specific study on gut microbiome and host interactions. I've chosen actually the insight into cell cycling regulation from the research of Sarkis K. Mazmanian. He and his team set out to investigate how bacteria colonize and set up long-term residence. Firstly, we're going to talk about the role of gut microbiota in host cell cycling. And the first topics we're going to focus on are Lactobacillus and Bacteroides. Lactobacillus is a genus of bacteria commonly found in the gut microbiome, which is known for its probiotic properties and beneficial effects on human health. This bacteria influences cell cycling processes in the intestinal epithelium through several mechanisms. So it influences cell cycling by producing short-chain fatty acids, which promote proliferation and differentiation in the cell. It modulates immune responses, which reduces inflammation and supports epithelial turnovers. It maintains epithelial barrier functions, and it induces antimicrobial peptides, which aid in microbial balance and reduce inflammation-associated damages. Bacteroids are a genus of bacteria as well. They are commonly found in the microbiota of humans and other animals. They are gram-negative, anaerobic bacteria known for their diverse metabolic capabilities and their ability to ferment a wide range of alkaloids. Bacteroids are common gut bacteria that, again, similar to Lactobacillus, produce short-chain fatty acids and modulate immune responses. They also signal through metabolites like polysaccharides A to regulate immune functions. Then we move on to talk about microbial-derived metabolites such as butyrate, propionate, and acetate, and how they play a crucial role in regulating whole cell cycling machinery within the gut. So these short-chain fatty acids, SCFAs, are produced by gut microbiota through the fermentation of dietary fibers, and they serve as key signaling molecules that influence various aspects of cell cycling in the intestinal epithelium. So butyrate is a cell studied known for its inflammatory and epigenetic regulatory properties. Propionate, on the other hand, is produced by gut microbiota and it influences cell cycling process in the intestine. While its specific effects on cell cycling are less well understood compared to butyrate, propionate has been implicated in regulating immune responses and epithelial barrier functions, both of which are closely linked to cell cycling dynamics. Acetate, in the other hand, is the most abundant SCFA in the gut because it plays a multi-phase role in the host microbiota interactions. The direct effects on cell cycling machinery are not exactly extensively studied. Acetate contributes to energy metabolisms a lot, mucosal integrity, and immune modulations, which collectively impact cell cycling in the intestinal epithelium. Overall, the role of gut microbiota-derived metabolites in the regulating host cell cycling is crucial for maintaining intestinal homeostasis and barrier functions. The gut microbiota is now considered as one of the key elements contributing in the regulation of the whole cell as one. So it is recognized as a pivotal determinant of host health, with its intricate interactions influencing various physiological processes. It maintains a symbiotic relationship between the gut microbiota and the host, which makes it very essential. And this is done due to several key reasons, which is metabolic activities, immunomodulation, epithelial barrier function, and nutrient absorption and energy metabolism. So, in conclusion, maintaining this relationship between the gut microbiota and the host is crucial for optimal cell cycling regulation and gut health. This symbiosis ensures continuous SCFA production, immune homeostasis, epithelial barrier integrity, and efficient nutrient utilization. All of this, which are essential for maintaining homeostasis and the health the body needs. Next, we're going to talk about the emerging techniques for studying gut microbiome host interactions. First, I would like to talk about agnotobiotic mice, also known as germ-free mice. There are invaluable tools in exploring the intricate interplay between the microbiome and the host physiology. These mice are raised in completely sterile environments. They are devoid of any microorganisms, which enables researchers to introduce specific microbial communities or pathogens to investigate their effects on various aspects of the host health and disease. By colonizing germ-free mice with defined microbial consortia or individual bacterial strains, scientists can actually elucidate the roles of these microorganisms in modulating host immune responses, gut barrier functions, and metabolic processes. So additionally, the development of humanized gnotobiotic mouse models, where human gut microbiota from maybe fecal samples or microbial communities are transplanted into germ-free mice. This allows for the study of human-derived microbial communities' impact on host physiology and disease susceptibility. So alongside colonization studies, advanced imaging techniques such as confocal microscopy and multiphoton microscopy provide real-time visualization and tracking of interactions between gut microbiomes and host tissues. Furthermore, advancements in multi-omics approaches, including many of them, have revolutionized our ability to comprehensively characterize microbial host interactions at the molecular level. These techniques enable researchers to analyze gene expression profiles, protein expression patterns, and metabolite profiles of gut microbes, which shed light on the functional activities, microbial responses to host signals, and the environmental cues within the gut ecosystem. Integrating these multi-omics approaches offers a holistic understanding of gut microbiome dynamics, identifying key biomarkers and therapeutic targets for gut-related disorders. As we move on, the impact of dysbiosis on gut cell cycling disease, we have numerous studies that have highlighted the association between dysbiosis, characterized by an imbalance in the gut microbiota composition, and various gastrointestinal disorders, including obesity and inflammatory bowel disease, or differently known as IBD. We specifically will be focusing in IBD. So in individuals with inflammatory bowel disease, this biosis disrupts the delicate balance of microbial communities within the gut, leading to alterations in microbial diversity, abundance, and functions. The inflammation in IBD disrupts gut cell cycling dynamics, which leads to aberrant proliferation and differentiation of intestinal epithelial cells. This dysregulation in cell cycling processes can result in compromised integrity, increased intestinal permeability, and mucosal damage, exacerbating the severity and progression of IBD. Moreover, dysbiotic microbiota may produce pro-inflammatory metabolites and virulence factors that further exacerbate gut inflammation and disrupt the host microbe interaction. So, in summary, dysbiosis plays a significant role in the pathogenesis of IBD by disrupting the gut cell cycling dynamics and promoting intestinal inflammation and epithelial barrier dysfunction. Understanding the impact of dysbiosis on gut cell cycling and disease progression in IBD is crucial for developing targeted therapeutic interventions to prevent this. As shown in figure 1, in very simple terms, when certain molecules like ATP and DMPs are detected by receptors on the intestinal cells, it triggers a process where proteins assemble into a complex called the NLRP3's inflammasome. This complex, when activates enzymes that produce inflammatory signals as like ILs, and different types of ILs. While IL-18 helps intestinal cells grow too much of it can trigger immune cells to overreact, which causes the inflammation. So the activation of this inflammasome can be both protective and harmful to the gut, depending on the actual circumstances. Lastly, we're going to talk about a specific study on gut microbiome host interactions. I've chosen to spoke about Sarkis Basmanian, and the theme of it is unraveling the gut microbiome. So, who was Sarkis Basmanian? He was a microbiologist initially studying at UCLA and later at Harvard and Caltech. He delved into the realm of gut microbiome research and uncovered the crucial role gut bacteria play in maintaining health. His investigation in IBD, inflammatory bowel disease, led to the discovery of a specific gut bacterium called Bacteroides fragilis, which potently anti-inflammatory properties have been very important. This finding challenged the conventional belief that all bacteria are harmful, suggesting that some bacteria have therapeutic potential for treating IBD by leveraging beneficial bacteria in this case. Additionally, Mazmanian's team identified previously unstudied genes responsible for bacterial colonization in the gut. Understanding these mechanisms offers insights into restoring microbial balance, which is often disrupted in various intestinal and inflammatory diseases. Expanding his research beyond the gut, Mazmanian explored the intriguing connection between gut microbes and neurological disorders like autism and multiple sclerosis. Through innovative experimentation, such as administering Bacteroides fragilis to mice with neurological symptoms, he unveiled promising links between gut microbiota and brain health. So, in essence, Mazmanian's research and bold hypotheses are shaping our understanding of microbiota and paving the way for novel therapeutic interventions. And I would like to conclude my project by mentioning a quote from Mesmanian, where he says that being intrepid and daring and fearless in his research results in the ability to ask questions that could be game changers, and that applies to every research you do. Because at the end of the day, having intellectual freedom is priceless. We follow our own interest, our own intuition, and our own curiosity. And I think it's through this type of innovative thinking that scientists like Mesmanian work to advance science and improve health. And I thought it was very motivating for anyone who wants to go into further research. And thank you for listening so much to my presentation.